This is the first video in the taxonomy video series. In this video we'll be covering the different divisions in taxonomy as well as viruses and prokaryotes. We'll get to archaea and eukaryotes later. So if you've had questions about the differences between cnidarians and platyhelminthes or pseudocoelomates and coelomates, we'll get to those divisions in future videos. On the left you can see I've written the major taxonomic divisions from largest domain to smallest species. There's a really simple mnemonic device to remember these. You've probably heard it before. It's dumb kings play chess on fine green sand. That's dumb kings play chess on fine green sand. So, and again, that goes from largest to smallest. So that means that two organisms of the same species are more closely related than two organisms of the same phylum. We'll talk more about that in the future as well. Really quickly, before we talk about the different domains of life, we're going to talk about something that's kind of debated whether it's living or not. That's viruses. So viruses basically they look like this. That's how they're always represented. This outer shell right here is a protein coat known as a capsid. Inside the capsid, the virus is going to contain its genetic material, either DNA or RNA. That shouldn't say RNA, it should just say RNA. It'll contain either its DNA or RNA, but never both, which is important. If the virus contains RNA, then it's going to also need to contain the protein reverse transcriptase. So the way a virus works is it injects its genetic material into a living cell, and then uses the living cell's reproductive mechanisms to reproduce its own genes, and then spread those to other cells. So if a virus infects a cell with RNA, then it's going to need to use reverse transcriptase to convert that RNA back into DNA that can be reproduced by the cell and then spread to other cells. So we've already talked about this. They're obligate parasites, meaning that they can't reproduce their own genetic material. They have to use a living cell to do that. They pass on genetic material through a process known as transduction. That's an important term to know. So transduction simply means when a virus passes its genetic material to a different cell. The lysogenic phase of a virus is also known as the dormant phase. In the lysogenic phase, a virus infects a cell, and the DNA or RNA that the virus passes on to that cell incorporates into the cell's genome and causes no noticeable effect in the cell. When the cell replicates its genome and divides, then the virus has spread to two cells now because the original cell that was infected divides. Eventually, some stimulus like UV light will cause the lysogenic viral DNA to become lytic or active. In the lytic phase, the viral DNA uses the cell to replicate and create thousands of copies of the virus, which then break the cell or lyse it and infect other cells. Lastly, these are important terms to know, just the differences between them. A virus infects eukaryotic organisms, so we've been talking about viruses, while a bacteriophage infects bacteria. So a bacteriophage is a virus that infects bacteria. They're actually the most numerous of any organism on the earth. Even though they're not really considered living, bacteriophages are the most numerous of anything on earth. So if a virus has infected a cell, it becomes a provirus. If a bacteriophage has infected a bacteria, it becomes a prophage. So pretty simple, but it's important to know those distinctions. Then we're going to cover prokaryotes. That's what the remainder of this video will be about. Prokaryotes are the oldest type of cells. When you hear the word prokaryote, you can basically just think bacteria. Like I said, they're the oldest type of cells. The oldest fossils of prokaryotes, I think, are 3.5 billion years old or so. Prokaryotes do not have a nucleus, so their DNA is circular, first of all, and it's also free-floating. It's not contained in a nucleus, as it is in eukaryotes. They also have no membrane-bound organelles, so their organelles are free-floating as well. That includes their ribosomes. You can see over here I've got the ribosomes breakdown for a prokaryote. They have a 50S large subunit, a 30S small subunit, and when they come together that makes a 70S ribosome. This isn't actual math. Obviously 50 and 30 don't add up to 70. These numbers just denote where the subunit or ribosome falls when centrifuged. So I just wanted to explain that. Prokaryotes also do not have histones. Histones are proteins around which DNA coils, as in eukaryotes and archaea. So they do not have histones, and they have ester fatty acid linkages. If you've looked at a fatty acid before, a eukaryotic fatty acid, it contains a glycerol. I'm not going to draw the whole thing. This would be the glycerol connected to an ester and then a carbon chain. So here you can see we've got an ester. That's an ester fatty acid linkage, which eukaryotes also contain. We'll talk about that with archaea as well. So prokaryotes and eukaryotes have that in common. They both have ester fatty acid linkages. Now, one thing that's unique to prokaryotes is their peptidoglycan cell wall. So peptidoglycan, you can tell from the word peptide, it's a mixture of peptides and sugar, glycans, glucose. So we have peptidoglycan cell wall. You'll often hear bacteria described as either gram-negative or gram-positive. What that means is when you're trying to visualize bacteria, you can treat them with a gram stain. If a bacteria remains purple after being gram stained, that makes it gram-positive, or that means it's gram-positive, and if it does not remain purple, then it's gram-negative. The reason a gram-positive bacteria remains purple after a gram stain is because they have a thick peptidoglycan wall that retains the stain, whereas gram-negative have a thin peptidoglycan wall that do not retain the stain. Gram-positive bacteria also have a single membrane, whereas gram-negative have a double membrane. So gram-negative bacteria have a membrane, and then their peptidoglycan cell wall, and then another membrane, whereas gram-positive only have a membrane inside their cell wall. You also may be asked on the DAT about the different toxins contained in gram-positive or gram-negative bacteria. You don't really need to know what the toxins do. Most of them cause illness in organisms. Gram-positive bacteria only contain exotoxins, while gram-negative contain exotoxins as well as endotoxins, which are also known as LPS or lipopolysaccharides. Those are contained in the outer membrane of the gram-negative bacteria. You can remember that they're in the membrane because LPS, lipopolysaccharide, is lipophilic, so they'll be contained in the outer membrane. Those are released when the gram-negative bacteria die. Moving on to nomenclature, you may be asked about this on the DAT as well. These are going to sound familiar to you. So if you see bacteria that are in a chain, the connotation for chain is strepto in bacteria. And you can also see these bacteria are spherical. The word for that is cocci or coccus. So these are streptococci bacteria. If the bacteria are clustered, then the prefix will be staph or staphil. So these are clustered and spherical, so staphylococcus bacteria here. If the bacteria, rather than being spherical, are bar-shaped, then it's bacillus or bacillus. So these are connected in a chain, and they are bar-shaped. So you can think about what their name would be. Moving on to conjugation. Conjugation is basically the closest bacteria will ever come to having sex. Normally bacteria reproduce through binary fission, which means that the bacteria reproduces its genetic material and its organelles and then divides its cytoplasm equally. So the two daughter cells are identical to the parent cell, and they also have equal divisions of cytoplasm, which is important to know. So like I said, conjugation is the closest bacteria will ever come to having sex. Conjugation occurs when one bacteria passes genetic information to another bacteria. That's done through the use of a pilus. A pilus is like a little stick that protrudes out of some bacteria. The pilus can be used to stick to membranes as well as to transfer plasmids or circular pieces of DNA between bacteria. So if, let's say that this bacteria was resistant to some antibiotic and it wanted to pass that resistance on, it would do that by using its pilus. So the plasmid or the gene it's passing on will just kind of go out on this pilus and can be absorbed by the other bacteria. So that's how antibiotic resistance is conferred between bacteria. And again, that's conjugation, different from transduction, where a virus passes on genetic material. F plus is how you denote that a bacteria is able to produce a pilus that has the gene for producing a pilus. F minus means that that bacteria is unable to produce a pilus. Transformation is pretty straightforward. You should be familiar with it if you've taken a molecular biology class. Transformation is the process where we use heat or calcium chloride to cause a bacteria to uptake a plasmid that we want to incorporate into the bacteria. That's transformation, again, which is different from conjugation, which is different from transduction. You should know those differences. Lastly, bacteria or prokaryotes have flagella, or like little tentacles used for moving, you can imagine them as. Their flagella are made up of flagellin, a protein known as flagellin, which is different from eukaryotes whose flagella are made up of tubulin proteins. So not super important, but good to know. You may have a question on that. That's all for the first taxonomy video. We'll cover archaea, eukaryotes in the next video.